Hello and welcome back to the second part of enveloped positive stranded RNA virus. And today we're going to talk about the life cycle of the dengue virus. So the learning outcome, previously we discussed about the introductions of the enveloped positive stranded RNA virus and specifically the dengue disease. But today what we're going to look at is the life cycle of the dengue virus, where we're going to talk about the attachment, penetration, translation of viral protein, how the genome is replicated, and how they assemble into a virus and eventually release from the host cell. So let's look at the structure of the virus. Because the virus is an enveloped virus where it has a glycoprotein on the surface. So what we can see here is it enveloped glycoprotein is surrounded the whole virus. And the virus is actually, if you look carefully, you can see they are actually a dimers. Most of the virus envelope protein is a dimer on the surface of the cell. So it has domain 1, 2, 3, and fusion peptide. So domain 1, domain 2, domain 3, and the fusion peptide at the tips of the domain 2. Right? So let's look specifically to this glycoprotein. Why is it so important for us to look at this glycoprotein? So the envelope is shortened into E. The reason why we call it glycoprotein because this protein has been modified when it's being produced. The protein normally that is having a modification by additional of carbohydrate to the protein, we call it the glycoprotein. So the glycoprotein, this enveloped glycoprotein is very important because this is the protein that surrounded the whole surface of the virus, and it is very important for the attachment of the virus, and it also initiates the entry of the virus into the host cell. Why is it so important also? Because it is on the surface and it is also the site where the antibody target and binds to it and prevent it from entering the host cell and replicate. Right, so the size of this glycoprotein is approximately 55 kilodalton. Now this kilodalton is actually a unit that normally we use when you run the gel on electrophoresis. We call it SDS page, polyacrylamide gel electrophoresis, where you can have a size, then you can identify the size of the protein. So there are three distinct domains, like I mentioned earlier. You have domain 1, 2, and 3. Each of the domain has its own functions. Now it's very important when you learn biochemistry, especially on the structure of protein. So you learn that it's primary structures, where you know the amino acid sequence. Then second structure, where you learn that there is the alpha helix and beta sheet for the protein structures. Then tertiary, when the protein structure fold together, so it becomes a structures, and then you have quaternary, so a few tertiary structures that combine and become a globular. So it's very important for us to know that when you learn about the primary structures, which is similar to this is first amino acid all the way to 394 amino acid. Please remember that the primary structure is just a sequence, but when it falls into a structures, into a secondary, for example, what we can see here, the circle here is the alpha helix, and these are the beta sheet of the secondary structures. But when this structure fall together, become a tertiary structures. Now, it will never be followed exactly in the primary structures. For example, in this case, all right, the red color is the N terminal. So we start with the N terminal here. So what we can see here is N terminal, they go move, and then when it comes to amino acid 52, it started to move into the yellow domain or the yellow color. And then it move on, and then when it reach 132, it go back to the red color. And when it goes to 193, it come back to the second domain, the yellow color. Then it go back to the red color, and eventually it go into the green color, the blue color. All right. So what it means here is that doesn't mean that when it's N-terminal, it must be domain 1. When it's C-terminal, it must be domain 3, and so on and so forth. It depends on how the protein fold, so that they resemble region and region and region, which give them the domain 1, domain 2, and domain 3. So I hope you understand this, okay? So that you know that the domain is not based on the primary locations. Alright? Okay, so domain 1 is normally the center, where it carry the domain 1 and domain 3 and domain 2 together. And domain 2 has an internal fusion loop which is involved in the membrane fusion. In here, which is the orange color here, but if you look at the previous one, it's the green color here. Okay, you just change the orange, the green to the orange color here. Now this fusion protein or fusion peptide is highly hydrophobic. Okay, it's highly hydrophobic, and the role of this fusion peptide is it will make the virus to fuse into the host cell. Okay, alright, and it's also important in the dimerizations of the E protein. So you can see major portion of the domain 2 are related, linked to each other or bind to each other in the dimer. So domain 3 is the immunoglobulin domain, where it should be involved in the cell receptor binding. So domain 3 binding, domain 2 fusions and dimerizations. Domain 1 is just to hold them together. Alright, so the replications begin with the attachment of the virus to the host cell. Right? In poliovirus, we know that the receptor for poliovirus is called poliovirus receptor, or PVR. For dengue virus, the receptor for this dengue virus, it depends on the cell type. So we have dendritic cell, and we also have macrophage, which are the target cell for dengue virus. So for dendritic cell, the receptor is heparin sulfate, and it requires a cofactor which is C-type lectin. Alright, this is the receptor and cofactor that is important for the dengue virus to bind to it and enter the cell. Whereas for macrophage, the receptor is mannose receptors, and the cofactor is C-type lectin, similar to this, but it has a domain family 5A. Right? So to make your life easy, just remember the cofactor for both of them are C-type lectin, and for dendritic, receptor is heparin sulfate, and macrophage is mannose receptors. So what happened now is when the virus enveloped the domain 3 bind to the receptors, okay, together with the cofactors here. So what happened is it will induce a process called receptor-mediated endocytosis. Alright, so this is the standard way of the virus attached to the receptor. Alright, of course, in the last lecture, we also mentioned that antibody-dependent enhancement, the way of the virus go in with the help, go into the host cell with the help of the FC receptors. But this is the standard way of the virus attached to the receptors, and one that is attached to the receptors, it will induce something called receptor-mediated endocytosis. What you can see is the purple color is the membrane, and what you have is the virus will now induce endocytosis, where the endosome surrounded the virus inside the cytoplasm. So we know that when endosome go into, and when the endocytosis occur, endosome go into the cytoplasm, what happens is proton pump will now start pumping the H plus or the acid into the endosome to make the pH inside the endosome become lower. This is a standard mechanism where the host cell, the cell is trying to fight off any pathogens inside the cell. So as the proton pumping acid into endosome, it lower the pH. So what happened is when this pH is being lower, the envelope glycoprotein started to experience conformational changes. So what you see here is a smooth surface of the virus, and now it becomes spiky. Alright, the structure smooth structures, and now they become spiky. So once it becomes spiky, you can see the yellow color is exposed. Okay, it used to be flattened, and now it becomes spiky. And what happened? Remember the orange color? The orange color is now on the tips of the spike. So I mentioned just now that the orange color is the fusion peptide, and the fusion peptide is now exposed. And the role of fusion peptide is to fuse the virus with the membrane of the host cell. So this is exactly what happened. So we are looking at the structures here, and this is an envelope protein in dimer. So as the pH change, it caused the conformational changes, leading to a spiky shape of the E protein or envelope protein. And now the envelope protein has a fusion peptide at the tips. So I mentioned that the role of fusion peptide is to fuse the cell. And what happened here, because the fusion peptide is highly hydrophobic and lipid bilayer is for hydrophobic. So what happened here is the fusion peptide will now penetrate the membrane. So it's sort of like a spring that link the two membrane together. Our virus is an envelope virus, so it has a lipid bilayer. Our cell is having a membrane that's surrounding the cell, so both of them are lipid bilayer. But the difference now is that envelope has a spike that poke through the lipid bilayer, and when this happened, so it's sort of like creating this envelope protein like a spring. So imagine here, it poke into it, and what happened is that when you pull this, look at it as if a spring. So when you have two membrane and you have a protein that behave like a spring that poke through the cellular membrane, and this spring, after you pull them, they will tend to come back. So what happened now is these protein now tend to come back. The spring come back together, and what happened? The membrane of the cell and membrane of the virus getting closer and closer, and what happened is when you have two lipid that come too close together, they will start to fuse. So very simple as an experiment, what you can do is take a bowl of water, put 2 drops of oil, and try to pull them together, and eventually you will see the oil will then fuse together. This is exactly what happened. The oil on the cell membrane, the oil on the viral membrane, so eventually they fuse together, and what happened? Once they fuse, they will start release the capsid, and this capsid will then dissociate and release the RNA of the virus into the cytoplasm. Alright, so that is the mechanism of how the virus attached to the receptors, go endocytosis, right? Low pH induces conformational changes of the spike protein and penetrate through the cellular membrane and bring them close together, and eventually causes the fusions of both membrane and release the capsid that containing the RNA, and capsid then dissociated and release the RNA into the cytoplasm. And because the RNA of dengue virus is polystrand RNA, so like it is also like poliovirus, it is also a functional mRNA. So what happened to this polystrand RNA, the dengue virus RNA? It started to migrate, travel all the way to the ER, okay, endoplasmic reticulum. So we know that the rough ER contains a lot of ribosome on the surface. So this rough ER, the RNA virus RNA will come to the rough ER and now initiate the next step, which is the translation or the productions of viral protein. So we have finished the first step, attachment, second step, fusion, then come to the third step, productions of the viral protein. Now, we mentioned just now that dengue virus genome has a cap, right? Unlike Hep C, Hep C, and Dengue virus, all of them using IRS, which similar to poliovirus. So once you understand the poliovirus mechanism of producing their protein, then you understand for the rest of the three. For dengue virus, there is the RNA is capped. Fantastic. Similar to the host cell. So the host cell mRNA also capped, right? So now we know that the EIF4E is important for the virus replications. However, the genome is lacking of poly-A tail. It doesn't have poly-A tail in their genome. So if that is the case, how did the viral RNA be translated in the protein? We know from the previous lectures that is the poly-A is very important for the translations. How did the virus RNA be translated into protein? If they're using the same translation mechanism, how did they overcome the competition from the host cell? This is two questions. Let's look at the first question first. Alright, so the primary mechanism of dengue virus translation occur via cap dependence initiations. Instead, you need the cap where it will be recognized and bound by EIF4E. Fantastic. 4E binds to the cap and go back. So let's have a look at this structure again. So this is where the cap of the dengue virus, where it will bound by 4E, and this 4E will then bind to 4G and bring everything together to form the 43S mRNA complex. Before we move on to the next step, I also mentioned that there is a very important step for this translation to be enhanced, which is the poly-A tail, which will be bound by poly-A binding protein and bring it back forward, all right, such as here, all right, bring the the tail, the 3' end to the EIF4G and together bound by poly-A binding protein initiating interacting protein type 1 that close together to this complex before it's being translated. Alright, so now the problem is there is no poly-A tail in dengue virus genome. How does this dengue genome produce the viral protein? So let's look at the genome. So the blue one here is actually the coding regions that code for your capsid all the way to the non-structural protein 5. So this is the 3' end, the cap. This is the 5' end, the cap, and 3' end. Alright. So what we can see here, there is no A at the end. Alright. So scientists trying to understand like how does the dengue virus produce their RNA since there is no poly-A tail. So what they did is that they tried to do a mutations, remove this portion, remove this. These are called pseudoknot, right? So remove pseudoknot 1 and 2, and try to analyze whether this portion of the RNA is where the poly-A binding protein binds to, because eventually you want the poly-A binding protein to bind at the 3' end and bring the RNA forward. But now they can't see it, they can't see a poly-A. So they suspect maybe this is the stem loop, it's the one that's responsible. So they remove this. Maybe this one is the one that's responsible, they delete this portion, they delete these portions. And what they found is after they delete this portion, they still can see poly-A binding protein binds to the 3' end. So eventually when they slowly analyze, they found that there are some region here and here that contains a few A's sequence, alright, in this region. So what they propose is that poly-A binding protein is actually bind to these whole regions that contains pseudoknot 1 and 2, where there's A here and a few A's in sequence here that allow the poly-A binding protein to bind to it, which will then bring the whole RNA to EIF5G. So here we already explained like how does the poly-A binding binds to, and eventually once they bound, they go back to the standard translation system. So it's very important for you to understand the basic concept of how the eukaryotic cell produce their protein. Once you can understand that, you will be able to apply that knowledge into any steps along the way. For example, if there is no cap, then they use IRS. If there is no poly A tail, then this is the one that they use. But this is specifically for dengue virus, right? For other viruses, maybe you need to study in detail how the poly A binding binds to. Alright, so now we answered the first question, then come to the second question, like how does it compete? So for poliovirus, because they use IRS so that the 2A protein can cleave the EIF4G and terminal to prevent the 4E to bind to it. However, for dengue virus, they rely on the 4E to bind to their cap of the RNA. So how does it overcome the competitions? So for dengue virus, they do not disturb the virus, do not disrupt the whole cell protein synthesis. Instead, what they do is, remember when I said the RNA will travel to the rough ER, and we know rough ER contains a lot of ribosome. Okay, that is where, when the RNA is present there, they will start producing a lot of protein. So they don't even have to compete because there are plenty of ribosome there, and immediately they can start producing their viral protein on the host cell. So appear overcome competition for host cell machinery via subcellular localizations. That is where they located. All right, the process to ER-derived membrane structures and so on and so forth. All right, so let's have a look at how the protein is being produced. So ribosome will start synthesizing the protein, and what we can see here, N terminal, and then you have the C terminal here. So the translation start here and produce protein, and because it's on the rough ER, and we know rough ER has an internal membrane, and what you can see here is this brown part is actually facing the cytoplasm. This blue part is the one that facing inward of the rough ER. Okay, so meaning that this is the lumen, this is the membrane of the ER. Alright, so rough ER on the surface, this is where the cytoplasm is. Inwards, that is where the lumen is. Alright, so ribosome started to synthesize the polypeptide, and then when they see the red, which is the signal sequence, it will go in, then they will start synthesizing. They see the blue stop transfer, they will go back to cytoplasm, and red go back in. So what this signal means is to tell the protein where it should go to. For example, if it is supposed to be in the cytoplasm, it will be here. Then it's supposed to be in the lumen, it should be here. So what we see here is the capsid is located at the cytoplasm. Means that on the surface of the rough ER, you will see a lot of capsid protein. Alright, inside the lumen, what we can see is we have pre-M envelope as well as NS1 protein. So the rest of the protein are basically located at the cytoplasm. Alright, so clear? On the surface of the rough ER will be the capsid as well as NS3, NS5, and other non-structural protein. Okay, whereas in the lumen we have pre-M envelope, which is a structural protein that is important. Okay, so cleavage of this protein into individual protein is done by the peptidase or proteinase that present in the lumen of the rough ER. Whereas over this side will be cleaved by the protein that coded by the virus. Right, so now we have the cytoplasm side. We have NS3, NS5, which is the protease, and we also have the NS5, which is the polymerase. So NS5 is the polymerase for the virus. Now, protein translations occur and it's continue, and then you start to produce a lot of the protein on the rough ER. This is a diagram to show you the functions. So just now I mentioned the 3 is the protease that cut the protein into individual protein. It is also a RNA helicase and so on and so forth. But the most important will be number 5, NS5, which is the RNA polymerase. So it is also has a functions of methyltransferase. So this is the protein. This is a polymerase that cap the RNA of the virus during the productions or the replications. Alright. So whereas the rest are mainly on the processing. Right, processing of the 135, right? Moderator. Okay, so now we have protein. So we've finished step 3, which is productions of viral protein. Now come to step 4, which is replications of viral genome. So in poliovirus binding of 3CD to 5' untranslated region and 3' untranslated regions. So 5' it will just push away the PCBP, poly C binding protein, and 3' push away the poly A binding protein to prevent the translation to occur. So what about dengue virus? How does dengue virus stop the translation and tell the viral genome to start replicating? So in the case of dengue virus, it is the polymerase NS5 that after being translated, this is the NS5 that bind to the 5' stem loop A, the one that is located on the 5' end of the genome, okay, which is this one here. Alright, so the NS5 is the one that bind to this stem loop and stop the translations to occur and start the process of replication. So the transition dengue genome architecture circularizations, alright, induced by protein binding. Now, there's one very important point that during the replications, the RNA of the genome will also be circularized. Remember in poliovirus, you have 3CD that bind to 5' end, 3CD that bind to 3' end, then the RNA start to circularize. This will enhance the replications of the viral genome. So for NS5, they also need to circularize their RNA. However, they don't have like 3CD like as in poliovirus. So the NS5 is the one that just stopped the translation. How does the RNA fold or circularize itself? So it appears that there are 2 secondary structure or the sequence in the 5' end and 3' end that is complementary to each other. So what we can see here, we have 2 region here called conserved sequence, and region here is upstream of AUG regions. So conserved region, CS, UAR stands for upstream of AUG regions. Alright? So what we can see here is the conserved region has a sequence of UA, UCAA, UAU, GCUG. Whereas what we can see is there is also a sequence at the 3' end that is actually complement to the 5' end, which is AG, UU, AU, and so on. So what we can see is A is complement to U, GC, UA, UA, AU, UA, AU. So this sequence and this sequence is absolutely complement to each other. We also found that there is another region which is the 5' UAR over here, the one that labeled in pink, has this sequence, and we also noticed that the 3' end also have the sequence that is complement to each other. Complement to each other. So what happened here is because they are complementary to each other, when we enlarge here, so this sequence will now form a complement, and this will come into here and form complement to the 3' end. So when they are complement to each other, it sort of like band the whole RNA and complement to each other and lock them into a circular shape. Alright? So when you have the lecture notes, you'll be able to look at the sequence and you can start analyzing how this fits well in here, and this one fits well and complement to them. Then what we can see here is this whole thing will bend and become a scalar shape. So the mechanism of dengue virus replication, so it started with, because it's RNA, so RNA, positive RNA. So in order for them to produce more positive RNA, it required them to produce a template which is negative strain. So in here, the NS5, which is the polymerase, is the one that is important, that bind to the 3' end and start synthesizing, just like poliovirus, these directions. Alright, bind to the 3' end and start synthesizing, find and add the nucleotide all the way to the 5' end. So now what we are producing is a double-stranded RNA, or we call it a replicative form. Alright, so this replicative form of the RNA is double-stranded. So you have one strand that is 5' to 3', and you also have another strand 3' to 5'. Alright, 5' to 3' and 3' to 5'. So the one that will be used as a template to synthesize more positive strain will be the negative strain, the one 3' to 5'. Alright, so what we see here is when the double-stranded RNA is being produced, they are called the replicative form. Replicative form, and then it will start synthesizing more and more positive RNA, and this is then converted into replicative intermediate. So at one time, the double-stranded will have at least 5 strains of newly synthesized positive strain RNA that being produced. So this is how the RNA is being produced during the replications of the viral genome. So what happened to this positive strain RNA genome? So there are two. One is this positive strain RNA, because the locations that is being produced is at the cytoplasm site of the RAF-ER. Immediately, this positive strain RNA can be translated again to produce more viral protein, okay? Or it can now be encapsulated or assembled to produce a new virus. So there are two ways. One is the newly synthesized positive strain RNA can go back and start the productions of protein, viral protein, or it can go for viral assembly to form a virus. So let's look at how is the second one. How does the virus assemble? So I mentioned that the polymerase at the cytoplasm site, so you produce a lot of positive strain RNA. So now, assembly of the virus begin with the virus go into the capsid. So the capsid is also located at the cytoplasm site of the RAF-ER, on the surface of the RAF-ER. Okay, so you have capsid that's surrounding on the membrane of the RAF-ER. Now you have RNA. So when the RNA in contact with the capsid, it will induce the capsid to go into the lumen site of the RAF-ER. Okay, let me repeat. The capsid is located on the cytoplasm site of the RAF-ER, and when RNA is being replicated and produced, the RNA will be in contact with the capsid and induce the endocytosis, alright, but into the lumen site. Okay, but into the lumen site of the RAF-ER. So what do we have in the lumen site of the RAF-ER? We have PM and we also have E. We know that the virus surface is covered by envelope as well as the M protein, right, on the surface. And then the capsid site is where the cytoplasm site is where the capsid located. Right, so now what happened here? This green color is the RNA, so it touches the capsid and it will now go into the lumen site And when they go into the lumen site, this is where the envelope protein and the M3M protein is located, and immediately they form this virion that is immature. Right, so do you remember that I mentioned to you the matured virus has a smooth surface, whereas the immature one has a spiky surface? So the smooth one has 50 nanometer, whereas the immature one has 60 nanometers. So when the capsid bud into the lumen site of the virus, what you see here is actually an immature virus that has 60 nanometers. So once the capsid bud into it, and now we have the immature virion, and the immature virion is now in the rough ER. It will then go into Golgi apparatus through the trans-Golgi network and eventually go out to be released from the host cell. So unlike poliovirus, where it breaks the cell to release the virus, dengue virus just bud out from the host cell. So I'd like to show you something about this. This is at the lumen site of the rough ER, where the pH is about 7.0. So the spiky surface of the virus is due to pre-M, right? Precursor M protein together with the E. So what we see here, the M, pre-M is the blue color and the red color. Alright? So the functions of this is the pre-M is trying to protect the fusion peptide from undergoing a prematured fusion again. Because if this fusion peptide, the red color fusion peptide, is exposed, it will immediately put back into the membrane and doesn't allow the newly produced virus to release. So this virus will forever inside the cell. However, the pre-M is the one that covers the fusion peptide and prevent it to undergo self-fusion again. So in rough ER, just now I show you, it has to from rough ER go into Golgi apparatus or trans-Golgi network. So when the immature virus travel from rough ER go into trans-Golgi network, the pH started to change. And when this pH started to change, you can see the structures from spiky started to smoothen out, but the pre-M remain attached. Alright, remain attached on the surface. So this is not a totally smooth surface of the virus yet. Alright, so it is still immature. Right? Right. So during the transfer from trans-Golgi network from one layer to the next layer, there's an enzyme called furin. Now, furin is an enzyme that cut protein specifically at certain site. Right. So what it happened here is the pre-M will now be cleaved by furin. However, the pre-M, the blue color, remain attached. Okay, the blue color remain attached until when the virus is being released outside of the cell, the pre-M, the blue color, will now be released, whereas the M remain hidden inside the E. Right? So this is the peptide that protect the fusion peptide to prevent it from undergo premature fusion. So once it's being released outside from the cell, the peptide will be released. Now we have a smooth, matured virus. So we basically have covered the 5 steps, right, of the life cycle for dengue virus. We look at how the virus attach to the receptors for macrophage or for dendritic cell, then how they penetrate, how the endosome, endocytosis, endosome, pH change, make it spiky, and eventually fuse the virus. Then we look at how they overcome the translation process of not having a poly A tail, and then finally genomic replications, how they produce their genome, then assembly, and finally release from the host. That's it. Thank you.